Okay, this chapter is on cell structure and function, so hopefully you remember a lot of it from biology. Right? No chemistry stuff, just biology. So the basic structure of the cell, we have a plasma membrane, and they're made up of those phospholipids. And then there's the cytoplasm, which would be the fluid inside of the cell, and they would hold the organelles. And then there's the nucleus in the center of the cell.
Plasma membrane around.
Mm-hmm. Yes.
What's consisting of the lipids of the
Phospholipids. So lipids. Yes.
Yes.
Is that a quarter quiz question?
No,
Oh. Oh, I forgot to grade correctors. I will have that by tomorrow. Yep. Okay, functions of the cell. It's the basic unit of life. Some cells are specialized for protection and support. They may produce and secrete various molecules, like in your immune system. Movement, muscle cells allow your body to move. Communication, cells produce and receive electrical and chemical signals that they send to other cells. And those signals can cause other cells to produce substance in order to create a response. Cell metabolism and energy release, that chemicals reactions that occur in the cell are part of cell metabolism. And then reproduction and inheritance, each cell contains DNA in the nucleus, and then some cells are specialized into gametes. So the plasma membrane. Good. The plasma membrane separates the intracellular from the extracellular material. So intracellular would be inside the cell, extracellular is outside. It allows for selective passage of substances into and out of the cell because the cell membrane has a polar area and a nonpolar area. It's able to keep some substances out or allow some substances to pass through. And then there's also channel membranes that allow different ions and molecules to pass through. There is a charge difference, which is called a membrane potential, across the membrane by regulation of intracellular and extracellular ions. A lot of it deals with the potassium and sodium ion concentration inside and outside the cell. And that membrane potential can open and close different gates in order to allow different substances to come into or out of the cell. And normal conditions outside of the membrane is more positively charged compared to the inside because there's more sodium ions outside the cell. And then when the sodium-potassium pump works, or if there's a channel that's open, then those can rush inside and change the membrane potential. There's a glycocalyx, which is a combination of carbohydrates and lipids. So there's glycolipids and glycoproteins. So we're combining the carbohydrate and lipid on the glycolipid, and this would be these yellow chains. Those are the carbohydrates, and so the ones attached to the blue spheres, those are attached to the phospholipids, and they would be the glycolipids. The glycoproteins are attached to these big globulin things passing through the membrane. Those are the proteins. So if it's a carbohydrate attached to the protein, it would be a glycoprotein. Excuse me. And this model, or the structure with the proteins floating around in the membrane, is called the fluid mosaic model because the membrane is very fluid. Those proteins can move throughout the cell or the membrane, but they cannot flip. So if you look at my pin and it's moving along in the membrane, it can move in the membrane laterally this way, but it can't flip like this. Okay? So Oh. So if this is the protein, this is like the outside of the cell, the inside of the cell, they can move around like this, proteins, but it can't do this. It can't flip upside down. This is just a closer picture. We will learn all about these proteins that are in the membrane. You will get to know and love them. Okay, the membrane lipids, those are the phospholipids, and cholesterols will predominate. The phospholipids create a bilayer. There's polar heads facing water in the interior and exterior of the cell, and those are symbolized by the blue spheres on the picture. Those are the polar heads. And then there's nonpolar tails facing each other on the interior of the membrane. Now, they use the word hydrophilic and hydrophobic. What do you think those mean? Yep, hydrophobic would be scared of water. And what would hydrophilic? Like think of Philadelphia. What's that? The nickname for that city? No, not the football team. Like Philadelphia is the city of. You guys know? City of brotherly love. Never heard of that? So it's water loving. Philo is the Greek prefix or Greek word for loving. Yep. And water's polar, and so the polar part is hydrophilic, and then the hydrophobic, they're pointing into each other because it doesn't want to touch the water on the inside of the outside of the cell. I don't know if you remember from last chapter what we usually draw a phospholipid would look like this, or it has two, these are the fatty acids, and then this would be the phosphate. But it'd look kind of like this since it's a bilayer. And it would keep going. And if you put phospholipids, just phospholipids in water, they will automatically arrange themselves like this because of that, the polar and nonpolar sections. Cholesterols are interdispersed among the phospholipids, and the amount determines the fluid nature of the membrane. So the more cholesterol you have, the more rigid the plasma membrane. And then the fluid nature of the plasma membrane allows for distribution of the molecules within the membrane, allows for the proteins to move around. The phospholipids will automatically reassemble if the membrane's damaged in any way. And the membranes can fuse with each other. So let's say here's one membrane, and now it's going to fuse with this other membrane right here. So here's a second cell or membrane. This is how secretory vesicles work. We'll get to those. We do organelles. But these two, if they're coming together, these two layers, this layer and this layer, will kind of combine and kind of connect, and this will be the outside layer then, and then this layer, it will kind of just open up so that maybe that's not this good spot to show it. Fine. So let's say between these two it's going to open up. Basically what happens is we form a new, like here would be the outside, and here, and then these two would connect, and these two would connect. Does that kind of make sense? I don't know how to show it otherwise. Kind of like, okay, so these are the two membranes. They're going to come here, and they're going to combine, and then they open up to make a new one. But they're able to do that very easily because they're fluid. Does that kind of make sense? where you have an inside-outside on the inside and outside of your fingers. The outside still stay the outside, the inside still stays the inside. It just opens up. Same with if during mitosis, at the end you have telekinesis where the plasma membrane comes in and then it pinches off and you have 2 new cells, same idea. Okay? And then, well, the membranes confuse with each other. That's what we were just talking about. Okay, membrane proteins. There are either integral or intrinsic. They're the same word for the same thing. But they extend deep into the membrane, and they would extend usually from one surface, so from the outside to the inside, whereas peripheral or extrinsic just stay on one side of the membrane. They're usually on the outside, but it could be on either the outside or inside surface, but they don't go all the way through the membrane. The integral proteins, they form channels that different molecules can pass through. Now, they're proteins, so remember, the protein has these long chains, and there's hydrogen bonding occurring that helps them fold up on each other and gives them their specific shape that allows for different molecules. So their function depends on their 3D shape, their tertiary shape. The chemical characteristics, markers, so there can be different like receptors too. Markers would be like all your cells have a specialized marker on it so that your body knows it's your cell and they can recognize it. Attachment sites, maybe it's a gated protein channel, and so it needs to attach an ion to it first and then it opens up. It'll have a channel for the whatever ion or molecule is going through. It has a channel for it to pass through. It would have a receptor. It could be an enzyme. It could be an enzyme that's attached to the surface and would catalyze another reaction. Or it could be also a carrier. It has lots of different functions. So marker molecules are glycoproteins and glycolipids. We already talked about what they kind of look like. So there's a carbohydrate chain attached to a protein or a lipid. And this allows cells to identify one another or another molecule. This is what is responsible for the immune response. So your body would recognize a foreign cell because its marker is different than what your body's marker is. It's the recognition of oocyte by a sperm cell and intercellular communication. This is where, like, if you have a graft or an organ transplant, you can have rejection because they recognize those markers as not your own, so it can start to attack that organ or that graft. Attachment proteins, there's cadherins and integrins. Cadherins attach cells to other cells, and then integrins are integral proteins. What does integral protein mean? You guys remember? We just had it on like 2 slides ago. Right, it goes from the deep through the membranes. It usually goes from the outside to the inside. But they attach to extracellular molecules, and sometimes when they attach, then that creates some sort of response inside the cell that can have a contact with internal or intracellular molecules. Transport proteins. There are a lot of transport proteins. The hydrophilic region faces inward, and the hydrophobic region faces outward. Remember, with the phospholipid bilayer, this part right here, is that hydrophilic or hydrophobic? The heads. Hydrophilic. Because they're facing outwards, they're touching water. So they like water. This part right here in the middle would be hydrophobic. Because it's trying to get away from the water. They're pointing inside. There's no water in between there. Does that make sense? That's the phospholipid. So now if we look at the proteins, the hydrophilic region has to face inward. So if I have this protein now that goes in here, like this, if it's going to pass between this hydrophobic region, it has to be hydrophobic as well. So the hydrophobic has to be on the outside of that channel. It's kind of like a tube that would go through. The outside would have to be hydrophobic also, so it's able to pass. And then there's a hydrophilic region on the inside. So now molecules that are out floating around the water that can't get through the hydrophobic region normally, now they have a hydrophilic area to go through and pass through the hydrophobic region. Does that make sense? Yeah, it's simplified, it's like a tube. And the inside would be water loving, so it can allow all those molecules through. If a molecule is non-polar anyways, it can pass straight through. It doesn't need a channel to get through. There's some molecules that can go straight through. But the ones that can't, they need some sort of channel because they aren't able to pass through the hydrophobic region. So they need that hydrophilic region on the inside of the tube to get through into the cell. The channel proteins, carrier proteins, and ATP power ponds are the ones we're going to go through. Channel proteins. There are non-gated and gated ion channels. Non-gated means they're always open. It's responsible for the permeability of the plasma membrane to ions when the plasma membrane is at rest. So ions can go in and out of the cell at will. Gated ion channels, there's some sort of closing of the channel until there's a certain stimuli that opens it. And that stimuli can be either a ligand, which is basically another molecule that attaches to a small molecule that binds to the protein, the channel protein, which would open the gate and allow the ions to go through. Or it could be a voltage-gated ion. So when, remember we said outside the cell is normally positive, if that membrane potential is switched, then it could open the gate and then allow ions to get through. There's two ways it can be gated. Carrier proteins are also called transporters. So the channel protein doesn't change shape, it's just either closed or open, and then it just allows free movement of ions through. Carrier proteins will actually change shape, and so they're integral proteins that move ions from one side of the membrane to another. There's a specific binding site, and the protein will change shape to transport the ion or molecule and will resume. So here in this picture, there's a binding site right here that is specific to a certain molecule. So here's the molecule that's going to be transported. Once it attaches, this whole protein changes shape so that now it's closed to the outside and open to the inside and will release that protein. It's not just a tube that you can drop something through. And there's several forms. There's uniporters. Uni usually means one, right? So we're moving just one particle. And then the symporters and antiporters move two particles, but symporters, so if you have a symbiotic relationship, what does that mean? A symbiotic relationship? You remember from biology? No. Usually, if they're talking about, they're both have the same purpose. They have the same. They're going for the same goal. Like symbiotic relationship could be, what are those fish that swim next to sharks? Yeah. Yeah. Or aren't there birds that sit on rhinos' backs and they always eat? So they're both
Living in perfect
they both
harmony.
benefit from each other. Okay, so symporters, if you think of it that way, they're both moving in at the same time. They're moving together. They're moving in the same direction together. That's how I kind of remember it. They move. So if it's outside the cell, they're moving both particles outside, inside the cell, or vice versa. Antiporters would be opposite. So they're moving, one molecule is going from outside to inside, and one's going from inside to outside, moving in opposite directions. ATP powered transport requires ATP. What is ATP? What is it used for? Energy. It's our energy molecule. So it requires energy to move a lot of substances, and it allows you to accumulate substances. We haven't talked about the concentration gradient yet, but the concentration gradient in the ion channel, it will move from high concentration to low concentration. For an ATP-powered transport, they can build up ion concentration. So it can go from low to high and still keep putting ions into the higher concentration. The rate depends on the concentration of the substrate, meaning the molecule that it's moving, and the concentration of ATP. If you run out of ATP, that transport protein is shut off. Here's a picture of it. So ATP, when it breaks, that changes the shape and allows the molecule or ion to move. Once that breaks off, then the active site it changes back into the original shape and allows for another ion to attach. Receptor proteins. They're proteins or glycoproteins in the membrane with an exposed receptor sites that can attach to specific chemical signals and would act as intercellular communication. And it's specific, has a receptor site specific for a specific ligand, specific chemical signal. So it's kind of like a ligand-gated channel where it's going to attach to a specific ligand, but it's not a channel. It's not opening up a channel. And sometimes this is linked to a channel protein, and this would be like a ligand. This would be a ligand-gated channel. An example here is acetylcholine is a neurotransmitter. You guys know what a neurotransmitter is? It's from your nerves, and it's from your nerves to your muscles. It's what's going to produce a change in the membrane potential to cause a muscle contraction. So there's receptor proteins that are on the membrane of your muscle cells, and when this acetylcholine comes across the synapse, there's a little gap between your nerve and your muscle. When it comes across, it's going to attach to that channel protein, and it will change the shape and allow sodium to rush in then. So it opens or closes the channel based on whether or not the acetylcholine or whatever the chemical is. It will open or close that channel. Changes permeability of cell of some substances. So cystic fibrosis, there's a defect in genes that cause a defect in those channel proteins, so it may not allow for the receptor to bind to that chemical. Drugs are also used to alter membrane permeability through attachment to channel protein-like receptors. Some receptors are linked to a G protein complex, it's called. Basically, it's a receptor protein that's attached to, this is called the G protein. There's 3 parts to this protein. And when the receptor attaches, it gets the signal, and then it would send this G protein to go have some sort of function inside the cell. like a secondary response. The receptor changes the channel of this protein, but that protein then would send this other protein off. This is just showing. We have 3 parts to it. You don't need to know the names of them. But when the chemical signal binds, then this would bind to it, and one of the parts breaks off and goes and has some sort of response. Stimulates the cell response. Okay, enzymes. You guys know what enzymes are, right? How do enzymes work? How do they speed up a reaction? Yes, they lower the activation energy. So some will catalyze reaction inside or outside the plasma membrane. The surface cells of the small intestine is an example where it has an enzyme here that breaks down a dipeptide into the individual amino acids. So it breaks that bond, the peptide bond. Okay, and that gives us all of the proteins we just talked about. Oh, I don't know. We were going to go through movement. Let me see how much we were going to go through. We can stop there today. We'll go over the movement.